The skin, see here we're going to see a very light skin is coming out. We're not going to use the first part, we will throw this away, okay? But the second part, here you see it's very thin. It's written on the board, can anyone tell me? Methylene, right. Now we're going to add few drops over here. See, it's taken. White glove again. Alright, see? Is it running? No. So it's ready. Now we have to place it over here. These are the prepared slides. We're keeping all the prepared slides here. We will view one by one. These two were already prepared earlier. Now, by turning it backwards, it goes up that way. You have to do it on your own, remember. So, we're going to place it in such a way that this specimen is directly below the lens, this objective lens. Okay? Now, let's turn on the light. By viewing through the eyepiece, at first everything is totally blurred. Now, by viewing through the eyepiece, we have to focus. The focus is like destroyed. You have to refocus again. So, while focusing, when you see that it's being focused now, then move very slightly. Like now you can view. See, you can see the nucleus very clearly now. Can you see the blue round? Yes, that is the nucleus. Cells are being focused very clearly now. One by one, the session you want to do first? And do. Prepare the slide, which we will be doing. Okay, do you think you can do it? Let's start. Here is the slide. Take one slide. You need nails for this actually. Add in both the plates. Okay. Switch it on. Yes. Now focus. While focusing, reacting, so we call this reagent or specimen. Then write iodine solution.